Hello everyone, welcome to today's webinar hosted by PC Genome. Today's topic is about lipid nanoparticle approaches in vivo CAR T therapies. Let's start. So what is CAR T cells? CAR T cells mimic natural T cells to recognize and attack infected or abnormal cells. In nature T cells, that recognition occurs through T cell receptors that binds to specific antigen presented by other cells. CAR T therapy enhances this process by T cell gene engineering to express chimeric antigen receptors (CARs), which directly target specific proteins or antigens on cancer cells. CAR T cell therapy is an innovative cancer treatment that modifies a patient's T cell to better fight cancer. It involves extracting T cells from the patient's blood, then genetically engineering them outside the body or ex vivo to express chimeric antigen receptors (CARs) that recognize specific protein on cancer cells. After modification, the CAR T cells are multiplied and infused back to the patient. This therapy is particularly effective against certain blood cancers, such as leukemia and lymphoma. Currently, there are 6 CAR T therapies have been approved by FDA in the United States. However, current CAR T cells involve complex and expensive process. The price for CAR T therapies are between $400,000 to $500,000 per dose in the United States. Patients also need to receive lymphodepletion, a short course of chemotherapy to kill original T cells before engraftment of CAR T cells. This process will increase risks of side effects. So can we bypass the complicated and expensive manufacturing process of traditional CAR T? Yes, here comes to the concept of in vivo CAR T, which engineer T cells inside the body using the advanced gene delivery tools, lipid nanoparticles, LNPs, or targeted vivo vector. In 2022, researchers from UPenn published a science paper about approach in vivo CAR T cells engineered to treat cardiac injuries in mice. The research group generated a mRNA encoding CAR designed against fibroblast active protein FAP. The function mRNA was packaged in CD5 targeted LNPs. CD5 is naturally expressed on T cell. With CD5 targeting feature, it can improve the endocytosis and expression of delivered genes in T cells. The following animal study also show the in vivo generation of transient FAP CAR T cells and cardiac function improvement after cardiac injury. Another research group from UPenn also focused on in vivo CAR Ts. They utilized their own ionizable lipid C14-4 to prepare LNPs for T-cell engineer. Data show that C14-4 LNPs have a delivery bias to spleen over liver. C14-4 LNPs were further modified anti-CD3 and anti-CD7 antibody fragment for additional targeting and endocytosis benefits. This platform successfully generates transient CAR T cells against B-cell cancer and show dose-dependent cytokine elevation, which suggests possibility of repeat dosing. Besides T cells, other immune cells can also be engineered to express CAR using lipid nanoparticle approach. The example here is the NK cells. HomeMap, a biotech company in Bay Area, used precision process system to generate a novel CAR mRNA LNP to effectively transfect NK cells expanded from primary PBMCs and to generate functional CAR NK cells. CD19 CAR mRNA and BCMA CAR mRNA were embedded into LNPs that result in 78% and 95% CAR expression in NK cells. BCMA CAR NK and CD19 CAR NK show significant high cytotoxicity in brain gamma granzyme B secretion compared with normal NK cells. After years of research and development, in vivo CAR T cells are on the verge of commercialization. A recent review article in Nature highlights studies that demonstrate the safety and efficacy of in vivo CAR T cells therapies for phase 1 clinical trials and preclinical models. The first example is CPTX-2309, which was developed by Capstan Therapeutics. It is an in vivo anti-CD CAR mRNA product delivered by a CD8-targeted LNP. In the preclinical studies, CPTX-2309 effectively engineered CD8-positive T cells from autoimmune disease patients to highly express anti-CD CAR and rapidly remove primary B cells. This CD8-targeted LNP model can also encapsulate CD20 CAR mRNA payload. It successfully delivers a therapeutic anti-CD20 CAR mRNA payload to T cells in non-human primate model in vivo. It successfully in vivo delivers a therapeutic anti-CD20 CAR mRNA payload to non-human primate model, resulting in transient generation of functional CAR T cells and depletion of B cells. This may allow broaden the application of CAR technology to B-cell-involved autoimmune disease through in vivo approaches. MTC02, developed by Myeloid Therapeutics, is the first LNP-assisted in vivo CAR immune cells therapy entered phase 1 clinical trials. The company has demonstrated CAR activity in human cells, follow systematic mRNA LNP delivery in mouse and non-human primate models. MT302 is targeting TROP2 positive epithelial metastasis. Using microfluidic mixing technology, researchers can conveniently produce nucleic acid encapsulated LNPs in a robust and reliable way. Precigenome provides a whole solution lipid nanoparticle preparation to cover different stages of the pipeline development, from early discovery, preclinical to commercial production. FlexS and FlexS Plus can prepare ultra-small batch size down to 100 microliter, which can save a lot of reagent and cost in the screen stage. FlexS Plus is a fully automatic workstation allows high-throughput screening, which allows synthetic 96 LNP samples within 1 hour. FlexMM Pro provide reliable and flexible LNP synthesis to meet the needs of preclinical animal studies. Precigenome also provides cGMP-grade model mass for commercial production. For more information, please visit us at www.precigenome.com. Thank you for your attention. I'll see you next time.